Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Stress Disrupts Reward in Mice, Insights into Neurocircuits and Molecular Targets in Psychiatry. My name is Lawrence Howes, and I'll be moderating today's presentation. I'm delighted to be joined by our guest speaker, Professor Christopher Pryce, Professor of Neurobiology and Biological Psychiatry at the University of Zurich. This webinar will explore the effects of chronic stress on reward behavior and associated activity in glutamate neuron pathway from basal amygdala to nucleus accumbens. Following the presentation, we'll have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these to the box to the right of your screen. Without further delay, I'd like to hand over to our speaker, Christopher, and I'd like to thank them again for presenting for us today.
Hello, my name is Christopher Pryce from the Psychiatric University Hospital and the University of Zurich, and it's my pleasure to talk to you about our research into how stress disrupts reward in mice to provide insights into neurocircuits and molecular targets in psychiatry. Working with mice, we focus in particular on an environmental manipulation called chronic social stress, which leads to changes in reward behavior and behavior towards aversive stimuli in mice. And what we see is that this chronic social stress manipulation leads to a decrease in reward learning and reward motivation, and an increase in learning about aversive stimuli and responding to aversive stimuli. We combine these behavioral studies with a variety of in vivo and ex vivo methods with which to study the central nervous system. And by combining neurobiological and research questions, we're able to obtain a complex picture and a comprehensive picture of how chronic social stress is acting on various pathways within the central nervous system in order to lead to these changes in emotional behavior. And with such models, using exactly the same models, we're then able to investigate the effects of compounds with respect to either the ability to reverse the effects of chronic social stress on reward behavior, that is to say, increase learning about reward and increase motivation for reward. And on the other hand, we're able to use these models to investigate the effects of compounds on, with respect to their ability to reverse the effects of the chronic social stress on the aversive system. In this talk, we will focus on the reward behavior and the reward neurobiology. And I won't go into the aversive system or into any of our pharmacological studies. But please bear in mind that this is the ultimate reason of why we're conducting this research, is to contribute to the much needed discovery and validation of novel treatments for psychiatric disorders. Of course, there is quite a large body of evidence with respect to those neural circuits of aversion and reward processing. And from this review paper by Luthi and Luscher, taking the schematic, the brain schematic from that paper, we can see some of the major brain regions and some of the neurotransmitter pathways with respect to long-range neurons that are connecting those various brain regions. And needless to say, a lot of these neurons are long-range projecting glutamate neurons or GABA projecting neurons or interneurons. And then there are brain regions, specific brain regions containing a relatively small number of neuronal cell bodies, such as the ventral tegmental area and dopamine, which has a major neuromodulatory effect on the activity of the same neurons that are receiving projections from glutamate neurons or GABA neurons. Here we see the amygdala, and here we see the nucleus accumbens. And it's this pathway between the amygdala and the nucleus accumbens which is the focus of the work that I'm going to talk about in this presentation. These are primarily long-range projecting glutamate neurons in the basolateral amygdala and projecting to the core and the shell of the nucleus accumbens. So here we see a schematic of the amygdala with its lateral nucleus, central nucleus, basomédial nucleus, and its basal nucleus. And in this talk, we're particularly interested in the glutamate neurons in the basal nucleus that are projecting to the nucleus accumbens. This pathway from the basal nucleus to the nucleus accumbens has been the subject of quite intensive research, but the picture is still rather unclear with respect to the valence, the emotional valence that these basal amygdala to nucleus accumbens neurons are responding to. In this schematic, we're seeing that we see the picture indicated whereby glutamate neurons in the basolateral amygdala are projecting either to ventral hippocampus or to the central amygdala or to the nucleus accumbens. And whereas here it's stated that these basal amygdala to nucleus accumbens neurons are primarily involved in positive valence processing, that is to say reward processing, there are other studies also conducted in mice which indicates that some of these neurons are also involved in negative valence information processing, that is to say aversion processing. So it's still an extremely interesting and intensive research subject as to whether the basal amygdala to nucleus accumbens pathway is a reward neuron pathway, an aversion pathway, or a combination of both pathways. And then of course we have the question, are these neurons are they monovalent? Are they neurons specific for reward processing and neurons specific for aversion processing? Or are there neurons which are responsible for both reward and aversion processing? Our initial approach when starting to look at this issue in my laboratory was to use retrograde tracing with the cholera toxin beta retrograde tracer conjugated with a fluorophore in order to allow ex vivo visualization of those cell bodies which have taken up the cholera toxin beta, abbreviated as CTB, into the neuronal cell bodies. So we're injecting the retrograde tracer into the nucleus accumbens and then allowing it sufficient time to be taken up into the cell bodies of all of those neurons which are extending axon terminals into the injection region into the nucleus accumbens core and shell. What we then did is we exposed the mice to either a social reward in the form of an estrus female mouse. So these were studies conducted in male mice. So the social reward was either a estrus female mouse, an estrus female mouse, and the social aversion was an aggressive a dominant mouse, which would there would be a period of distal exposure with the two mice separated by a barrier, then a short period of proximal exposure where the mouse injected with the retrograde tracer was briefly attacked by the aggressive dominant mouse, and then the two were separated again. And then the control mice were left undisturbed and were not exposed to any stimulus. We then perfused and sectioned the brains across the entire extent of the amygdala. So from Bregma level -1 up to Bregma level -2.6, which is pretty much the entire anterior posterior extent of the amygdala. And then using confocal microscopy and immunostaining for the immediate early gene protein CFOS, we were able to visualize both the CTB positive neurons in these basal amygdala sections and the CFOS positive neurons in those same sections where CFOS was taken as an indicator of responsiveness to either social reward or social aversion. And then we looked at the coexistence of these two stainings of both CTB and CFOS within the same neuronal cell body to tell us which of these CFOS positive neurons were projecting to the nucleus accumbens. We found a characteristic profile of the CTB positive BA-NAc neurons, a relatively small number of neurons in the very anterior part of the basal amygdala. And then in this intermediate region of the basal amygdala, that's where we see the highest number of BA-NAc BA neurons projecting to the nucleus accumbens. And these profiles look similar when we take the density of the neurons. Now, when it comes to looking at the number of neurons that were expressing CFOS in these BA cells, not necessarily BA neurons projecting to the nucleus accumbens, but all BA cells that were identified in the sections, then we can see that both for social reward exposed mice and social aversion exposed mice, there was a much higher number of CFOS positive cells than was the case in the no stimulus mice. And we can see that these profiles are very similar for those mice exposed to reward and those mice exposed to aversion. And what we then did, of course, was to investigate which of these CFOS positive neurons were neurons in the basal amygdala projecting to the nucleus accumbens. And here again, we can see that in the intermediate basal amygdala, there were a similar number of neurons that had responded either to social reward or to social aversion. And this in the intermediate basal amygdala, we have a relatively high number of such neurons of both of those groups, both social reward responsive and social aversion responsive. And whereas when we go into the posterior basal amygdala, we see that there is a reduction in the reward responsive neurons, we see that there's a relatively high number now of social aversion responsive neurons in the posterior basal nucleus. And here we have the same data, but now with the amygdala divided into anterior, intermediate, and posterior. And we see that in the intermediate basal amygdala, we have this relatively high number of both social reward responsive neurons and social aversion responsive neurons. And then in the posterior basal amygdala, we see that we have relatively less social reward responsive neurons and more social aversion responsive neurons. What we don't see in this figure, but what we've been able to establish in another experiment, is that the majority of these neurons in the basal amygdala, in the intermediate basal amygdala, projecting to the nucleus accumbens, are monovalent with respect to social reward and social aversion. So we can't say that they're monovalent in that they only respond to reward or only respond to aversion, but at least as with regard to these two highly salient social stimuli, the majority of these neurons in the basal amygdala, intermediate basal amygdala, projecting to the nucleus accumbens, either respond to social reward or to social aversion, but not to both stimuli. We wanted then to see if we could obtain some further evidence that that the BA to NAC neurons in the intermediate part of the BA were different to those neurons in the posterior part of the BA. So we were able to establish that the relative numbers of social reward and social aversion responsive neurons were different in these two regions, and we wanted to see if that was reflected in any other parameter that we could measure. And so what we did in order to investigate that was to again inject retrograde tracer, the cholera toxin beta, conjugated with fluorophore 555, injected that into the nucleus accumbens, and then allowed it to be expressed, and then collected the brains, this time rinsing the blood from the brains with buffer and not fixing the brains, in order that they would be compatible with laser capture microdissection and collection of, in order to collect cell bodies which were CTB labeled in the basal amygdala, and then extract the RNA from those cell bodies and then use RNA sequencing to look at the transcriptome of these basal amygdala to nucleus accumbens neurons and compare them in the intermediate BA and the posterior BA. So we're injecting CTB. After some weeks, we are then collecting the brains, rinsing them of blood, and then we're sectioning the brain at the in those we're sectioning the brain coronally in order to collect 10 micrometer thick sections, which are then being placed onto special membrane slides in order that they would then be compatible with laser capture microdissection. So this here we're looking at some of these sections from the images taken with the laser capture microdissection system which was the cell cut system of molecular machines and industries, and we were using the appropriate channel in order to allow us to visualize the CTB in these sections, and using the cell cut tools at a diameter of 35 micrometers, we were identifying those regions in the basal amygdala which were CTB positive. And we're assuming that these CTB positive regions that could be visualized were indeed the cell bodies of neurons in the basal amygdala projecting to the nucleus accumbens. And we collected from each mouse in this experiment, we collected 500 such pieces of tissue from the intermediate basal amygdala and 500 from the posterior basal amygdala. We pooled those 500 samples per mouse, and then we extracted the RNA from those 500 pieces of tissue per mouse, and that RNA then went through amplification and library preparation and RNA sequencing using the Illumina system. And what we firstly wanted to establish was to use marker genes for various cell types in order to hopefully verify that we were indeed collecting primarily tissue from glutamate neurons. And so we can see here that we have the neuronal marker SNAP25 highly expressed, and the glutamate neuronal marker SLC17A7 also highly expressed, whereas marker genes for GABA neurons, for astrocytes, for oligodendrocytes and the precursor cells, and from microglia, all demonstrated extremely low levels of expression, confirming that we had indeed collected primarily tissue from glutamate neurons, and because this tissue was expressing CTB, these were glutamate neurons that were projecting to the nucleus accumbens. When we then compared the transcriptomes of these glutamates of these basal amygdala to nucleus accumbens neurons, we were able to identify a really substantial separation of the two subregions of the basal amygdala. So there were some 430 genes that were upregulated in the intermediate basal amygdala compared to the posterior basal amygdala, nucleus accumbens projecting neurons, and there were some 350 genes which were downregulated in the intermediate basal amygdala relative to the posterior basal amygdala. So out of the approximately 13,000 genes that were expressed in these samples, about 900 of those genes were differentially expressed between the two regions, and it was possible to obtain a complete separation of these two populations using cluster analysis based on the 500 most differentially expressed genes in the sample. So clearly then, the intermediate basal amygdala and the posterior basal amygdala, with respect to the glutamate neurons that are projecting to the nucleus accumbens, have a very different transcriptomic profile. In addition to them, in addition to those neurons being different proportions of social reward responsive and social aversion responsive neurons. And so we can see that the technique of laser capture microdissection and RNA sequencing was very useful here for providing additional information with respect to these two populations of BA-NAc neurons being indeed rather different in their transcriptomic profiles, as well as their emotional responsiveness. Moving on to some behavioral data now. What we're looking at here is an experiment that shows you how chronic social stress reduces reward learning. And I'll start off by explaining a bit more about the chronic social stress procedure. So it's a 15-day procedure where the male black 6 mouse is maintained next to a dominant aggressive CD1 mouse, and the two are separated by a perforated divider in the cage. And for a short period per day, the two mice are placed in the same compartment, and the black 6 mouse is threatened and attacked by the CD1 mouse, and we time the periods of physical attack so that these are for each of the 15 days there is 30 to 60 seconds of physical attack. And so a very short period of physical attack to ensure that there is no wounding of the black 6 mice, and then the two mice are separated again and maintained either side of the divider. And on each of the 15 days of the procedure, the black 6 mouse is placed next to a different CD1 mouse. So there is an element of stress uncontrollability to this procedure, in that the black 6 mice are not able to control the aggressive behavior of the CD1 mice. And there's also an element of unpredictability in that the identity of the aggressive dominant mouse changes across days. The reward test that I'm going to show you here is called discriminative reward learning and memory. So what it involves is the presentation of a tone. This becomes a discriminative stimulus, because when the tone is playing, and only when the tone is playing, is the mouse able to obtain a sweet tasting, rewarding sucrose pellet when it makes a nose poke response into the feeder. So the tone, the discriminative stimulus tone, plays for a maximum of 30 seconds. If the mouse makes just one response into the feeder during that 30 seconds, the tone will stop and a sugar pellet will be delivered. There's then an interval of between 20 and 80 seconds, and then the next tone will play. And if we look at the number of chocolate pellets that the mice obtain, we see that this is that the chronic social stress leads to the mice obtaining fewer of these chocolate-flavored sucrose pellets that are being delivered as a reward. And we give the mice 3 tests on 3 consecutive days after the chronic social stress procedure has completed. If we look at the response latency of the mice, that is to say the time between the onset of the tone and the mouse making its response, we see that that latency is higher in the stress mice than it is in the control mice. And we can see that particularly in the control mice, they're getting faster to respond to the tone as they go across tests. And this is indicating that they're learning the relationship between this tone and the availability of a pellet if they make a nose poke into the feeder. If we look at the average interval between consecutive responses during the intertrial intervals, we see that that is also higher in the chronic social stress mice compared to the control mice. And now if we calculate the learning ratio, which is the response interval divided by the DS response latency, we see that whereas this learning ratio increases across tests, as the control mice become quicker in their response to the tone, it stays at around 1 in the chronic social stress mice. So demonstrating that the chronic social stress mice have a deficit in their learning about the relationship between the tone and the availability of the rewarding chocolate-flavored sucrose pellets. So what we then did in the next experiment is that we took that same model of chronic social stress and the discriminative reward learning and memory test, and we combined the model with fiber photometry. So we now targeted the BA-NAc neurons in that intermediate region where we had demonstrated that there was a relatively high number of reward responsive neurons in the BA projecting to the NAC. Of course, as you will remember, there was also a relatively high number of aversion responsive neurons in that same intermediate region. But we nonetheless hypothesized that if the BA-NAc pathway is contributing to behavior in this test, then we might also expect to be able to observe a decrease in the activity of these neurons in the chronic social stress mice relative to the control mice when the discriminative stimulus is presented. So if we look first again at the behavioral data, we see that the behavior was similar to that observed in the previous experiment, in that the stress mice were obtaining fewer rewarding pellets than the control mice. The response latency to the tone was higher in the stress mice compared to the control mice, and it was only in the control mice that we see this decrease in the response latency as we go across tests. Again, we see that the ITI response interval was also higher in the stress mice than in the control mice. And again, we see that by the time we get to the third test, there is a significantly lower learning ratio in the chronic social stress mice than in the control mice. Now, whilst this behavior was ongoing, we were also conducting fiber photometry. So we were using a calcium sensor, GCAMP6, which fluoresces, and that fluorescence fluoresces in response to calcium intake into the neurons, and that fluorescence is detected using the optic fiber implanted directly above the region into which the GCAMP6 viral vector had been injected. And what we could see is that during this DS on phase, that is to say, during the phase when the tone was playing, whereas in the control mice we can see an increase in the GCAMP6 signal relative to baseline, which is the dashed line here. In the chronic social stress mice, the activity of the BA-NAc neurons did not change during the presentation of the discriminative stimulus. So this reduction in the responsiveness to the discriminative stimulus in terms of behavior was coincident with a decrease in the responsiveness of the BA-NAc neurons to the discriminative stimulus. And what we can see here is the mean value of the calcium signal across this DS on phase per mouse. And we can see that there is a significant decrease in that activity in the stress mice compared to the control mice. Now, immediately afterwards, as they make their feeder response, then we can see that again, immediately after the feeder response, this is the point at which the sugar pellet is delivered, we can see that the activity of the BA-NAc neurons is relatively low in the stress mice compared to the control mice. And then whereas when the pellet is delivered and the mice are eating the sucrose pellet, whereas the activity of the neurons gradually and consistently decreases in the control mice, it increases in the stress mice. So both during the DS on phase and this so-called post-discriminative stimulus sucrose phase, we can see that there are effects of the chronic social stress on these neurons in the intermediate basal amygdala, projecting to the nucleus accumbens. What we then did was to ask, well, what happens if we specifically target these BA-NAc neurons in the intermediate basal amygdala? And what we did here to target those neurons was to inhibit them by using a viral vector system, which resulted in the expression of tetanus toxin light chain in the basal amygdala to nucleus accumbens neurons, which has the effect of cleaving the protein VAMP2, which is essential for normal glutamate release from these BA-NAc glutamate neurons. So we were inhibiting the BA-NAc neurons by having them express tetanus toxin light chain, which results in a cleavage of the VAMP2 protein, and therefore a reduction of glutamate release from these neurons, projecting specifically to the nucleus accumbens. And what we could see that whereas there was now no significant reduction in the number of chocolate pellets eaten, there was a higher a trend to a higher DS response latency in those mice expressing tetanus toxin light chain relative to the two control groups. And when we look at the learning ratio, we can see that similarly to the chronic social stress mice, there was a reduction in the learning ratio in the mice expressing tetanus toxin light chain, specifically in the BA-NAc neurons relative to the two control groups. And here we're looking at the average across the 4 days of testing, and seeing that relative to these two control groups, those mice expressing tetanus toxin light chain in their BA-NAc neurons are, similarly to chronic social stress mice, less able to learn the relationship between the tone and the availability of the sucrose reward. So some evidence then that BA-NAc neurons are indeed contributing to reward-directed behavior, suggesting that this pathway, which is in which these neurons are showing different activity after chronic social stress, suggesting that that reduction in responsiveness to the stimulus that predicts reward and to the reward itself is contributing to this deficit in reward learning. What we then wanted to do was to validate that the VAMP2, that the tetanus toxin light chain had indeed functioned as it should do. And in order to do this, we turned again to laser capture microdissection and RNA sequencing using the MMI cell cut system. So now we were identifying those neurons which were expressing EGFP, which by necessity needed to be BA-NAc neurons. We were sectioning those brains from these from the tetanus toxin light chain group and one of the control groups, and we were then using now the EGFP, which was expressed alongside the tetanus toxin light chain, to identify the BA-NAc neurons, collecting populations of 500 such neurons per mouse or per brain, and then running RNA sequencing. Again, we were able to show that the neuronal marker SNAP25 was highly expressed, as was SLC17A7 in the control mice, but we saw a significant reduction in the mice that were expressing tetanus toxin light chain, and we saw a corresponding increase in the microglia marker. There were a large number of genes which were upregulated in the tetanus toxin group versus the control group, and many of these genes were involved in pathways for cellular senescence, apoptosis, and chemokine signaling. And so putting that information together, it looks like what happens is that in the BA-NAc neurons expressing tetanus toxin light chain and not releasing the glutamate, we get a buildup of glutamate, which has an excitotoxic effect on those neurons, and probably also stimulates activation of neighboring microglia. So we were also collecting some microglia tissue as we were collecting the neurons. So here, using laser capture microdissection in order to validate the efficacy and understand the effects of what the tetanus toxin light chain is doing in those neurons that expressed it. As a final experiment, we used laser capture microdissection and RNA sequencing to look for the effects of the chronic social stress on gene expression in the BA-NAc neurons. Again, the method of injecting cholera-toxin beta, allowing the mice to recover, allowing the uptake of the tracer, and then exposing the mice to chronic social stress, collecting those brains, collecting the CTB labeled neuronal cell bodies in the BA, and then running RNA sequencing. And here we identified only a moderate number of genes being differentially expressed in the chronic social stress mice compared to the control mice. The majority of these neurons were involved in pathways of glutamate signaling, and therefore suggesting that chronic social stress impacts on the signaling function of these BA-NAc neurons. But what we need to remember is that there are two populations of neurons in the intermediate BA that are projecting to the nucleus accumbens. It's not only reward neurons, but also aversion neurons. And we think that this might be one reason why we see a relatively small number of differentially expressed genes in these BA-NAc neurons after chronic social stress. And what we really need to do is to have a method of identifying specifically the BA-NAc reward neurons and to differentiate them from the BA-NAc aversion neurons. And that's what's indicated here. And we're using the method of targeted recombination in active populations in order to identify which neurons have been are responsive. For example, using a CFOS-dependent TD tomato expression, using the TD tomato to identify those BA-NAc neurons which are responsive to a reward, such as an estrus female, as we used in the first experiment, and then again using laser capture microdissection to now sample those reward positive neurons, using a similar method to sample aversion responsive neurons, and then comparing those two populations in terms of the transcriptome to identify hopefully genes which are only expressed in reward neurons or aversion neurons, and those genes will then be used for strategies for targeting the reward pathway or the aversion pathway specifically. And this will be another application of the laser capture microdissection system. So this work was primarily done in the laboratory by Hannes Siegrist, Julia Poggi, Alexandra Greta, Lorraine Madur, Diana Kukalova, Christian Inaiken, Giorgio Bergamini, and a lot of the work that we do is in collaboration with Boehringer Ingelheim, and is also funded by the Swiss National Science Foundation. And thank you very much for your attention, and I look forward to your questions.
Thank you very much for that informative presentation, Christopher. It is now time for our question and answer session. So if you have any further questions, please do send those in. You can submit these by typing into the questions box to the right of your screen. So we've had a couple of questions in today. To start, what is the minimal number of neuron samples that need to be pulled to yield sufficient RNA for RNA sequencing?
Oh, that's a very good question. Thank you very much. So as I explained, we've used populations of 500 neurons, and even using 500 neurons when it comes to quantifying the integrity of the RNA. It's typically below the limits of the fragment analyzer to tell us what the integrity of the RNA actually is. But when we go through amplification and library preparation and RNA sequencing, then we see that we are detecting somewhere between 12,000 and 13,000 genes in a reproducible and reliable way, and in a way where using cell type specific marker genes, we're able to validate that we have indeed been collecting tissue primarily from the targeted glutamate neurons. We haven't tried with less than 500 because it's frozen tissue. I'm sure that it's not compatible with single cell RNA-seq, but I could well imagine that using less than 500 neurons, if the methods that people want to use doesn't allow them to collect 500 cells because their region of interest is too small, or the population of the cells of interest is too small, then I would be quite confident that it should work with probably 250 cells as well. I'm pretty sure that during pilot studies we did try 300 neurons, and it gave us results very comparable to 500 neurons. So I hope that provides some helpful feedback to that very good question.
Excellent. Thank you for clarifying that one. We've got another question in here. How long can tissue be stored, and how long can histological sections be stored prior to LCM?
Tissue, if the brain is after the brain is collected, if it's immediately frozen, then it can be stored pretty much indefinitely at -80 Celsius. We always rinse perform a perfusion on the anesthetized mouse, so deeply anesthetize the mouse with a pentobarbital, check for the pain reflex, and then perfuse the mouse, or check that there is no pain reflexes, perfuse the mouse with a buffer in order to flush out the blood so that there is no contamination of the transcriptome with blood, and then can then proceed to almost indefinitely freeze your brain samples at -80, or when you then go on to prepare your sections using the MMI slides, then we've stored those sections for up to 7 days, and we haven't seen any detrimental effect of that storage. Again, freeze the sections. We haven't seen any detrimental effect of keeping the sections under those mounted on the slides, 10 micrometer thick sections mounted on the MMI slides and frozen, and then thawed and performing the laser capture microdissection, and you will still obtain good cells. What we like to do is to collect the sections, and within 1 to 2 days to process those sections, rather than collecting a whole batch of sections on the cryostat and then several days later proceeding to collect the cells from those many sections. We tend to work with a few sections at a time to make sure that the interval between obtaining the sections and obtaining the tissue from the sections is as short as possible, but we have gone up to 7 days.
Great, thank you for that. On to the next question here. Is it possible to use 2 different fluorophores to identify 2 cellular populations that coexist in the same brain region?
Another super question. It is possible, as long as those fluorophores are sufficiently different. So you can imagine it similar to a confocal microscope. If you've got two secondary antibody fluorophores, they need to be sufficiently different that they can be detected by different channels, and it's exactly the same as on the cell cut MMI laser captured microdissection system. So you need your two fluorophores that you're working with to be within the range of two of the channels, and then you can happily differentiate them and collect from two different cell populations very cleanly without any cross contamination. We're running 4 different channels on our cell cut system, and works very well in terms of collecting 2 different fluorophores.
Great, thank you, Christopher. We've got another question that's come in. Could you elaborate further on the ethical aspect, please?
Oh, it's another really super question. It's absolutely important that when you're doing such animal model research, that you're working with valid animal models, and there's various ways to assess the validity of the model to make sure that you really are performing experiments with animals which are relevant to the human disease for which you want to improve understanding, and for which you want to identify new pharmacological targets and then validate those targets. And I'm very happy to say that we've been able to demonstrate that our models are valid in this respect. So based on validation data that we've obtained with our models, and said that this target and this compound is efficacious in the model, human studies have then been done. So firstly, proof of concept studies in healthy humans, and then trials in patients. And indeed, we've been able to predict the effect of the compound from the results of our models. So we really have sort of prospective predictive validity evidence from our models, and this is absolutely essential in order to ethically justify the inclusion of animals in such research, that the models have validity with respect to making accurate predictions about the relevance of those pathways and receptors in the human brain, and also their involvement in causal involvement in psychiatric disorder, and therefore their potential as a target for new treatments. And we really need new treatments in psychiatry. Stress-related disorders are extremely common throughout the world, and the first line of treatments is still to target the serotonin system, which we've known for some 70 years to be useful in some patients, but not useful at all in other patients. And so this is really an area of unmet medical need, where the cost-benefit analysis of the animal experiments, and the cost to the animals and the benefit to the humans really allows for the justification of these very carefully performed experiments with valid animal models. Thank you very much for that question.
Excellent. Thank you very much for clarifying that. So that is all we have time for today. I'd like to thank our expert speaker, Christopher, for the informative presentation and discussion, and a big thank you to everyone for joining us online. We hope you found this a worthwhile session. If you do have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with questions for our speaker. And remember, you can download related resources in the tab to the left of your screen, and this includes the Certificate of Attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it'll be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.